Hi guys, how's it going? Hello again. From yesterday to today, I got new glasses, Harry Potter style. Yay! So, if you didn't see part 1, I'm going through a past paper, and there's a link for the past paper and the mark scheme in the description. And I basically did half of it because it was taking quite a long time, and this is the second half. And we got up to, if you go on the link and you load up the documents, for the past paper, we got up to question number 5. That is on page 16 out of 36. And for the mark scheme, it got up to page 15 out of 24. So get that ready. And I had already read this question. So it's a question about blood clotting and the effect of smoking on these different proteins that take part in blood clotting. So there are a couple of tables, and one of them refers to this study where they took blood samples from men aged between 35 and 50 just after waking. That's non-smokers, smokers, and then also smokers after the first cigarette. And I mentioned how the sample size of 9, 10, and 10 is very small. And then looking at the mean thrombin concentration in arbitrary units, it's so much higher for smokers after their first cigarette, but it's also double for smokers before even smoking upon waking compared to non-smokers upon waking. Also, if you also notice, the standard deviation for the smokers after first cigarette, plus minus 76, is absolutely bloody massive. But it does fall in line with also the mean thrombin concentration being higher as well. So this would look good on a graph, better than in a table. Anyway, we've got the first question. Analyze the data to explain why smoking can increase the risk of blood clot formation. 4 marks. So analyze the data to explain why smoking can increase the blood. This is fairly straightforward. The data obviously refers to both tables. So in the first table, we have the number of cigarettes per day and the mean blood plasma concentration of fibrinogen. And then the second one is the mean thrombin concentration upon waking. So one is looking at how much you smoke every day. And then the second one is looking at how smoking affects the mean thrombin concentration, but also the effect after you stop smoking. So even after you've not smoked overnight, the effect is still there, right? So essentially, there is a correlation between smoking and the increase in blood plasma, fibrinogen, and thrombin, both of which are key to blood clotting. So if these things increase in the blood, then they make it more probable that you will get a blood clot. And therefore, smoking more is associated with this, right? So go back to the question. The questions are phrased to guide your answer. So it's not just to make you understand what they're talking about, but the actual words used. So for example, analyze the data. It's telling you to look at the table and say what's going on in the table. To explain means find a reason. So the explanation is about how thrombin and fibrinogen act in blood clotting as clotting factors, as proteins that physically create clotting, can increase the risk of blood clot formation, right? So 4 marks, that's basically what you're talking about. And in the mark scheme, we have 1 mark, increase in smoking leads to more thrombin and fibrinogen. Another mark, increases are significantly higher between no overlap over standard deviations. And another one, thrombin converts fibrinogen into fibrin, so this is the mechanism, and fibrin forms clots. So the one thing that I didn't mention is actually also including the standard deviation. So instead of just saying that it is higher or that it is different for smokers compared to non-smokers, also take into account the standard deviation, and then there's no overlap. So essentially, if this was a graph, you would see the standard deviations and you could see that they're overlapping or not overlapping. But because it's in a table, it's making it a bit more awkward, and actually you'd have to do a bit of calculation to kind of figure out whether or not it's overlapping, to be honest. For example, if you wanted to compare in the second table smokers after waking and smokers after first cigarette, you would take the number that is highest for smokers after waking, so that's the mean concentration, which is 121, plus the 47 standard deviation, so that is the uppermost level, that would give you 121 plus 47, 168, and then take the one for smokers after first cigarette, 365, and what is the lowest one for that one, because that one is higher. So the lowest level for that may overlap with the highest level for smokers after waking. So the lowest for 365 is 365 minus 76. So that would be 280 or something. And 280, or whatever it is much higher still than 168. So that's how you know they don't overlap. And since they don't overlap, that is considered a significant increase. So, moving on. If you don't get what I mean with the standard deviation, just try drawing it. So just try drawing the means or in Excel or somewhere and then putting the standard deviation above and below the mean. So essentially for 56, you'd have plus 1 to 57 and also minus 1 to 55. So your whole thing would be covered between 55 and 57. And for 121, it would be plus 47 and then minus 47, and that gives the whole range for where the deviation is. Right. The diagram below shows some of the cells found in blood. So we got some cells labeled Q, R, S, and it says which row of the table correctly identifies the cells labeled Q, R, and S. 1 mark. Options A, B, C, D. Erythrocyte, monocyte, neutrophil, neutrophil, lymphocyte, monocyte, monocyte, neutrophil, erythrocyte, etc., etc., etc. So essentially, what are these cells? Now, if you know the relative sizes of them, it helps. The white blood cells are much bigger than red blood cells, so also the red blood cells are much more prevalent than white blood cells. So most of the cells in this diagram are red blood cells, and you can see that S is a similar size and structure to the other one, so that must be an erythrocyte. So if S is an erythrocyte, then looking at the table of options, S being an erythrocyte is only option C. So you don't even really need to bother with Q and R. Is this correct? No. Fuck. I failed. It's actually D. Monocyte, neutrophil, lymphocyte. So Q is a monocyte, R is a neutrophil, and S is a lymphocyte. That's really confusing. I don't know, it looks like it could be a red blood cell. Also, I don't understand, see, it's confusing. This diagram is showing red blood cells as in the surface of red blood cells, but it's showing the white blood cells as a section of them. So essentially, this, I don't know, this doesn't look like what you would see under the microscope. Ah, sad. Very sad. So S is a lymphocyte, R is a neutrophil, and Q is a monocyte. So actually it has options C and D for monocyte and neutrophil, and then it's just a case of whether S is an erythrocyte or a lymphocyte. And it says it's a lymphocyte rather than an erythrocyte. And frankly, I don't know why. Oh, I do know why, of course. I think that might be maybe the nuclear material, potentially. Red blood cells don't have a nucleus. So if the darker area of the white blood cells is actually their genetic material, although it looks like there's a lot of it, and the lymphocyte would basically just be, I don't know, then that would explain why that would be a white blood cell, not a red blood cell. Let me just quickly check another diagram of it, just to clarify that. So yes, the lymphocyte does have a massive nucleus. So the darker region is the nucleus, and S, as you can see, is basically a nucleus with some tiny bit on the top. Yes, so therefore, it cannot be a red blood cell because red blood cells don't have a nucleus. So therefore, S is also a white blood cell, and lymphocytes have got a massive nucleus, therefore S is a lymphocyte. If you know that, then you don't even really need to know much about Q and R being monocytes and neutrophils. But basically the distribution of the nucleus inside the cell, and obviously the size of the cell, can help you get to the answer. It's confusing because that's a section, that's obviously a section through the cell, whereas the red blood cells are just the surface, so it makes no sense. But anyway, right, describe the function of a neutrophil in defense, 2 marks. So the neutrophil, which is in the diagram cell R. So for 2 marks, I guess you would say within the immune system, it acts to phagocytize, that's not a word, to undergo phagocytosis of invading agents. And phagocytosis, for 2 marks, you mention phagocytosis and just say how it works, which is essentially breaking down the material of the invading pathogen or agent. And the mark scheme says engulf/phagocytosis and digest or break down antigen or pathogen or bacteria or virus or microbe. So any combination of that. It says ignore kill, a bit too colloquial, and reject produce antibodies, because that's not what it does. It's a generalized cell. It's not a specific cell that produces antibodies. Accept correct references to lysosomes or phagosomes. Right. Not a very good start, but yeah, that's why you have to look at a lot of micrographs and things under the microscope to pick up the details. Question number 6. Gills are the site of gas exchange in fish. Ooh, I like gills. The diagram shows some of the gill lamellae from a bony fish. We've got blood entering the gill on one side, blood leaving the gill on the other side, and then the lamella in the middle. Draw 1 arrow on the diagram to show the direction of water flow across the lamellae. 1 mark. So across the lamellae, you would draw an arrow to show the direction of water flow. So essentially, is the water flowing in the same direction as the blood leaving the gill, or in the opposite direction of the blood entering the gill? So of course this is making reference to the countercurrent exchange system, the gas exchange that takes place in the fish gills. So the idea is that it's countercurrent, which means it's against the current. So obviously if the current of the blood entering the gill is going downwards, then the water will be going upwards. It's a cross. It shows a diagram, and it's a cross. Line drawn from left to right over or between 1 lamella. From left to right. So it's going basically across both. So it's going between them. So it's going from the blood leaving the gill to the blood entering the gill. So it's going from the oxygenated side to the deoxygenated side. That's confusing. That diagram is confusing. Anyway, the diagram shows... I'm just like, don't bother, just don't bother. The diagram shows a section through the head of a bony fish. The graph shows the pressure changes in the buccal cavity and opercular cavity during ventilation. So this is the fish head, and we've got the buccal cavity, opercular cavity, operculum, and then we've got a graph that shows pressure changes with time, and we've got the pressure in the buccal cavity and then the pressure in the opercular cavity. So it's kind of a weird graph. Anyway, it asks for 1 mark, the operculum will be closed between, and then it gives you options of what time. So the operculum will be closed between 0.4 and 0.5 seconds, where the pressure is at highest, 0 and 0.4, where it is increasing, 0.4 and 0.6, when it increases and then suddenly drops, or zero point 0.2 and 0.4, again, where it is increasing. So the operculum will be closed when? I would say between 0.4 and 0.6. It's just a guess. It's not, it's no, it's A. It's between 0.4 and 0.5 when the pressure is highest. So it's not when the pressure is decreasing, it's just when it's at the highest, full stop. So presumably it opens after that. I don't even know what the opercular cavity is. I've got no idea. Genuinely clueless. The table shows some information for 5 species of fish: toadfish, butterfish, sheep's head, mullet and mackerel. Activity level, surface area of gill lamellae, mass of fish, comparison of surface area to mass. You can sort of see where this is getting at. The ability, the anatomy of the fish to carry out more or less gas exchange depending on its activity level. So the comparison of surface area to mass of mullet is 1 mark, and it gives you some numbers: 1,000 centimeters squared per gram, 10 centimeters squared per gram, 1 centimeter squared per gram, or 0.99 centimeters squared per gram. So the comparison of surface area to mass of mullet. So we go with mullet. Mullet's got a high activity level, and we've got a blank in the table where the comparison of surface area to mass is. And we are given the surface area in millimeters squared, and we are given the mass of fish in grams. So the comparison is in centimeters squared per gram. So the mass in gram is fine, we can leave it as 250, but the surface area needs to be converted from millimeters squared to centimeters squared, and centimeters are bigger than millimeters, so we need to divide by 10, and because it's squared, we need to divide by 10 squared, which is 100, which gives 2,525, divided by 250, which is 10.1. So the answer is B. And the mark scheme says that the answer is B, so that's good. Analyze the data to explain the difference in the activity levels of these fish species. 3 marks. So analyze the data. We've already looked at the data. We look at the comparison of surface area to mass. You can very easily see that the low activity is 1.5, the medium activity is 4.6, and the high activity is 10-ish. So that's relatively straightforward. You can also see that the surface area of the gills alone isn't necessarily that useful, because the sheep's head, which only has medium activity level, has a higher surface area than the mackerel, which has a high activity level. That's why it's important to use the comparison of surface area to mass. And since it's referencing the activity level, we need to connect the availability of oxygen through the gills to the activity of the rest of the body, and in this case, the relevant part is the muscle. So for 3 marks, we have in the mark scheme, more active fish have higher surface area to mass ratio, which is the comparison column, therefore can absorb more oxygen for respiration for more muscle contraction. Right, water polluted with heavy metal ions can cause the death of fish. The photographs show cross-sections of the lamellae from a fish caught in unpolluted water and from a fish caught in water polluted with heavy metal ions. The first diagram, gill lamellae from a fish in unpolluted water. It's got a lot of white space, empty space, blank space. Gill lamellae from a fish in water polluted with heavy metal ions. A lot denser, the lamellae look like they're stuck together. Use the photographs to explain why fish die in water polluted with heavy metal ions. 2 marks. So making reference to the diagram where the gills, well, the gill lamellae look like they're stuck together, or they don't have much surface area, there's something wrong with them, which of course interferes with gas exchange. So in the mark scheme, again, gill lamellae are thicker or swollen or touching or less surface area or less contact with water / have decreased water flow, therefore less gas exchange or oxygen uptake. Right, moving on. There are currently only 3 surviving species of elephant. Asian elephant, Elephas maximus, African bush elephant, Loxodonta africana, and African forest elephant, Loxodonta cyclotis. Complete the table showing the classification of the African bush elephant. 2 marks. So we've got domain, kingdom, phylum, order, family, genus, species, and then some of them are filled in: Animalia, Chordata, Mammalia, Proboscidae, and Elephantidae. So the domain is eukaryota or eukaryote. We use our useful mnemonic for the different levels of classification. So D, domain, K, kingdom. Dirty, kinky policemen can often find gay sex. So can often find, can is C for class, so mammalia is class. And then genus and species is obviously the name, which for the African bush elephant is Loxodonta africana. So the genus is Loxodonta and the species is Africana. And the mark scheme confirms that, so that's straightforward. All African elephants used to be classified as a single species. Recent comparisons of African elephants demonstrated that they are two separate species. The diagrams show the appearance and locations of the two African elephant species. We've got the African bush elephant and the African forest elephant. On this grid in the background, that means that the scale is the same, so you can compare their size. And the question is, give one reason why scientists used to classify African elephants as a single species. One mark. They have a similar appearance. They have similar features. So the mark scheme says similar appearance or similar trunk or tusks, and they've not observed if they could breed together. It's a bit random. I don't see what that has to do with the diagram, but anyway. Scientists agreed to separate the African elephants into two species based on new evidence from DNA sequencing. State how scientists reach agreement on new evidence. One mark. So this is referring to peer review, where scientists review each other's work, and they review the evidence, and they try to reach a consensus if possible. Presenting at conferences, communicating, publishing. So the mark scheme says publish in journals / present at conference, or peer review, or writing papers, or other scientists repeating the work. Recently, scientists extracted mitochondrial DNA from a frozen Arctic mammoth. This DNA was sequenced and compared with the mitochondrial DNA from the Asian elephant and from the African bush elephant. The percentage similarities in the DNA sequences are shown in the table. So we got species, mammoth, Asian elephant, and African bush elephant, and the percentage similarity in DNA sequence. So for mammoth, it's 100% with mammoth because it's itself, and 98% with Asian elephant, and 96% with African bush elephant. Which diagram shows the correct evolutionary relationship for these three species based on the data in the table? One mark. So we have diagrams. The first one shows a common ancestor giving rise to all three species at the same time equally. The second one shows a common ancestor between the African elephant and the lineage that includes the mammoth and Asian elephant, making them more closely related than with the African elephant, and different combinations of these. So going back to the table, we can see that the mammoth is more closely related to the Asian elephant, and the Asian elephant conversely is more closely related to the mammoth compared to the African bush elephant. The African bush elephant is mostly related to the mammoth, but 96% rather than 98%, which is between the Asian elephant and the mammoth. Therefore, the most closely related species of the three are the mammoth with the Asian elephant. So the diagram that portrays that is B, where the mammoth and the Asian elephant are more closely related than with the African elephant. So that's B. Yep, okay, moving on. Scientists also investigated the effect of temperature on the percentage saturation of hemoglobin with oxygen using blood from an Asian elephant. The oxygen dissociation curves are shown in the graph. Hemoglobin, this should be a familiar graph. Hemoglobin saturation against pressure of oxygen in kilopascals at different temperatures. And of course this should make you think of the mammoths and how they lived in cold regions, and this is where this whole temperature oxygen dissociation thing is getting to. So you can see that the curve shifts to the right with increasing temperature, which means that with higher temperature you have for example, let's look at 4 kilopascals on the x-axis. For 10 degrees we have almost 70% saturation, and for 43 degrees we only have like 30% saturation. So essentially more oxygen becomes available at higher temperature. In the alveolar capillaries, the partial pressure of oxygen is 12 kilopascals at a temperature of 38 degrees, and hemoglobin is fully saturated with oxygen. In active muscle tissue, the partial pressure of oxygen is 2 kilopascals at a temperature of 43 degrees. Use the graph to determine the percentage saturation of hemoglobin in active muscle tissue. 1 mark. So it's given us a situation of active muscle tissue at 2 kilopascals for a temperature of 43 degrees. So if we look at 2 kilopascals for the dotted line of 43 degrees, we can match that on the y-axis to a hemoglobin saturation percentage of 10%. So the answer is 10%. When 1 gram of hemoglobin is fully saturated, it carries 1.3 centimeters cubed of oxygen. Calculate the volume of oxygen released by 1 gram of hemoglobin to this active muscle tissue. Show your work in 2 marks. So it might be tempted to think, okay, if the active muscle tissue hemoglobin saturation is 10%, then you just work out 10% of 1.3 centimeter cubes of oxygen. And that's what I was going to do initially. But if you think about it, the saturation of oxygen in hemoglobin is the amount of oxygen in the hemoglobin. So if it's in the hemoglobin, it's not in the active tissue. So actually, the volume of oxygen released to the active tissue is the oxygen not present in hemoglobin. So if you've got 10% saturation, that means that 90% of it is available to the tissue rather than in the hemoglobin. And therefore, you actually have to work out 90% of 1.3 centimeters cubed, which the mark scheme gives as 1.2 centimeters cubed. It makes sense because 1.2 centimeters cubed is almost 1.3, so it's 90%. So it makes sense. You know the calculation's right. Scientists extracted the genes for mammoth hemoglobin and used them to produce mammoth hemoglobin. The oxygen dissociation curve for mammoth hemoglobin at 38 degrees was found to be the same as for the Asian elephant at 38 degrees. Lowering the temperature did not shift the oxygen dissociation curve. Explain how these observations show that this hemoglobin enabled mammoths to be adapted for life in cold Arctic regions. 3 marks. So essentially, if the oxygen dissociation curve doesn't shift with lower temperature, it means that the oxygen is still available at that temperature, at a lower temperature, as much as it is at a higher temperature, enabling respiration to take place, aerobic respiration, and of course connecting that with keeping warm and surviving in the cold Arctic region. So we have affinity of hemoglobin for oxygen doesn't change, therefore oxygen is still released, therefore heat is still produced by respiration. Sucrose is synthesized and transported through plants. The diagram represents the mass flow hypothesis for sucrose transport in a plant. So we see, mass flow hypothesis, sink and source, sugar transported in phloem from source to sink, and water return in xylem from sink to source. So of course the source of sugar is the photosynthesizing leaves, and the sink is the non-photosynthesizing parts like the roots, whereas for water uptake, again, water is taken up through the roots, therefore that is where water is coming from, and it's being transported to the leaves, right? So which row in the table shows the conditions present in the sink and the source of a plant carrying out rapid photosynthesis? 1 mark. So in rapid photosynthesis, you have a lot of sucrose being produced in the source. So in the source, you would have a sucrose concentration that is high, and because the sucrose concentration is high and sucrose is water soluble, it will affect the water potential and it will lower the water potential. So we would have a high sucrose concentration and low water potential in the source. And in the sink, because the rapid photosynthesis takes place before the plant has a chance to actually transport these things, you would expect a relatively higher water potential there with a lower sucrose concentration. So that is option C, which is right. Okay, moving on. The effect of root temperature and oxygen on the movement of sucrose through a plant was investigated using the apparatus shown in the diagram. Plant leaves placed into a sealed glass chamber with an atmosphere consisting of radioactive carbon dioxide 14, and air. 3 small tubes, A, B, and C, 200 millimeters apart, penetrating the phloem vessels of the stem, and plant roots in a thermostatically controlled water bath. The water bath was set at 10 degrees, quite cold, and a solution from each of the small tubes was collected. The time taken for radioactive sucrose to travel the first 200 millimeters and the second 200 millimeters was recorded. The experiment was repeated at 20 degrees and 30 degrees. The whole experiment was then repeated, replacing the air with the nitrogen gas. So the air is mostly nitrogen gas anyway, but it also has oxygen and it also has non-radioactively labeled carbon dioxide. So in these experiments, that was removed and it was all just nitrogen. that displaces oxygen and carbon dioxide. The results are shown in the table. So we have the different combinations of different experiments. If we look at the mean rate of sucrose transport in the last column, we can already see that it's much lower for where the air was replaced with nitrogen. So calculate the mean rate of sucrose transport when the plant is kept in carbon dioxide, labeled carbon dioxide and air at 30 degrees. The mean rate of sucrose transport when the plant is kept. So that is at 30 degrees, so that's the missing bit in the table. And essentially, we can work out the mean first between 1.75 and 1.83, which the mark scheme says is, I think, 1.78. And then we can work out the mean rate of sucrose transport, which is 200 divided by this 1.79 rather than 1.78. So essentially, because the mean is given in for 200 millimeters an hour, and the mean rate is in millimeters an hour, which is 1 millimeter an hour, you divide 200 by the mean, which is 1.79, which gives 111.73. Which is the highest, very high, of course, because it's at 30 degrees. Analyze the data in the table to explain the effect of temperature and oxygen on sucrose transport. 4 marks. So the effective temperature, of course, is that it goes up, and the effect of oxygen is that it also goes up. Of course, oxygen is not included in the table, so you have to understand that where you have carbon dioxide with air, that air has oxygen. Where you have carbon dioxide with nitrogen, that no longer has oxygen. So you're comparing the air as the one containing oxygen. Of course, it's much faster, whereas where it doesn't, it slows down. So the availability of oxygen is connected to the ability to carry out aerobic respiration, which is involved in the transport. So essentially, there are multiple points here. Changing temperature has no to little effect in nitrogen, or no oxygen, not in air. Increasing temperature increases rate of transport because temperature affects enzyme activity. Oxygen increases the rate of transport because the translocation is an active process or requires respiration. Before the 20th century, tigers were once widespread across the whole of South and Southeast Asia. Over the last 100 years, their geographical distribution has been reduced by over 90%. The diagram shows the current distribution of 3 varieties of tiger. So we're shown a region of South and Southeast Asia, the Bay of Bengal, the Indian Ocean, Sumatra, and we've got a key showing Sumatran tigers in grey, obviously mostly in Sumatra, Indo-Chinese tigers, and Bengal tigers. Use the map to explain how the different varieties of tiger may eventually become separate species. 4 marks. So the map clearly shows that these species are there's a word for this. I don't remember the word. It shows that the different species are prevalent in these separate areas, and how some of these areas, for example, the Indo-Chinese tiger territory is actually separate from the Sumatran territory by water. So that's kind of a natural barrier there, and there doesn't seem to be a lot of crossing over between the different species. So if they stay geographically isolated, over time they will acquire different adaptations that may mean that they can no longer interbreed. So then they would become separate species. So the mark scheme says allopatric speciation would occur because tigers become geographically isolated, so that they are reproductively isolated. They're no longer going to breed. They may become genetically different or accumulate different mutations due to reduced inbreeding or more outbreeding. And that's actually it. I didn't think it was gonna end. That's it, right? I didn't know whether to redo the questions because I got more of them wrong this time than last time, and it's embarrassing actually. But you know what? It's real, and this is how learning happens. And there's no such thing as a perfect streak, and there's no such thing as perfect, and there's no such thing as clean and neat. This is how it is. This is what it is. And yeah, I mean, good luck, and see you next time. And I got loads of old pound coin going on. Okay, bye!